Welcome to this lecture on reproductive biology. Today, we are going to explore the fascinating and essential topic of contraceptive methods. We will uncover how these methods interact with the human body to prevent pregnancy, and we will learn the mathematical tools scientists use to measure exactly how effective they are. Let us dive in. To begin our journey, imagine a highly secure vault that opens only once a month. This vault represents the female egg. Now, imagine a team of tiny intruders, the sperm, trying to get inside. To protect the vault, you could build a physical wall, change the locks, or make the environment completely inhospitable. This is exactly how contraceptive methods work in the human body. They act as biological security systems designed to prevent the sperm from meeting and fertilizing the egg. By understanding this natural cycle, scientists have developed ways to safely pause reproduction. Now that we understand the basic analogy, let us look at how these methods actually intervene. Contraceptive methods, commonly known as birth control, halt conception at different critical stages of the reproductive process. Some methods stop the vault from opening at all by preventing the release of an egg during ovulation. Others physically block the sperm from reaching the egg to stop fertilization. Still others prevent a fertilized egg from attaching to the uterine lining during implantation. The choice of a specific method depends on various factors, including its efficacy, reversibility, and the individual's overall health profile. Building on these intervention strategies, we must recognize why this matters so much globally. The importance of contraceptive methods cannot be overstated. They are the absolute cornerstone of family planning and global reproductive health. Without these methods, individuals would have very little control over their reproductive destinies, leading to significantly higher rates of unintended pregnancies and maternal mortality. Contraception allows families to space out births, which drastically improves the physical health and survival rates of both mothers and infants. Furthermore, barrier methods like condoms serve a vital dual purpose by also protecting individuals against sexually transmitted infections. Understanding the vital role of contraception leads us to an important question. How do we know which methods work best? To evaluate exactly how well a contraceptive method works, scientists use a specific mathematical metric called the Pearl Index. This index measures the effectiveness of a birth control method by calculating how many pregnancies occur per 100 women using the method for one full year. Understanding this metric is crucial because it allows doctors and patients to compare vastly different methods on a single standardized scale, proving scientifically whether a new pill is actually better than a traditional barrier method. Now that we know what the Pearl Index is, let us look at its mathematical formula. The formula is R equals C times 1,200 divided by E. Conceptually, this scales the data from any clinical trial to a standard century of women months. The variable R represents the Pearl Index itself. The variable C stands for the total number of unintended pregnancies observed. The number 1,200 is a constant scaling factor, representing 12 months multiplied by 100 women. Finally, E represents the total months of exposure. If the number of pregnancies, C, increases, the Pearl Index, R, increases, meaning the method is less effective. Conversely, a higher exposure time, E, lowers the index. With our formula established, let us see this with a concrete example. A clinical trial is testing a new daily oral contraceptive pill. We are given that 200 women use the pill for exactly 6 months each, and 3 women become pregnant. We need to calculate the Pearl Index. First, we find the total exposure time E by multiplying the 200 women by the 6 months, giving us 1,200 months of exposure. Next, we substitute the 3 pregnancies and our exposure time into the formula. The 1,200 in the numerator and denominator cancel out. Our final answer is a Pearl Index of 3. Physically, this means if 100 women used this pill for a full year, exactly 3 would experience an unintended pregnancy.
Having seen a straightforward trial, let us evaluate a scenario involving human error. A study evaluates a barrier method under typical use conditions, where people might make mistakes. We are given 300 women using the method for 10 months, resulting in 15 pregnancies. We must calculate the typical use Pearl Index and compare it to the perfect use index of 2. First, we multiply 300 women by 10 months to get 3,000 months of total exposure. We plug 15 pregnancies and 3,000 exposure months into our equation. Multiplying 15 by the constant 1,200 gives 18,000. Dividing 18,000 by 3,000 yields a Pearl Index of 6. Physically, this shows that human error triples the failure rate from 2 to 6, highlighting the critical need for patient education.
Now that we have mathematically evaluated effectiveness, let us explore the actual biological mechanisms of these methods. We will start with the most direct approaches. Barrier methods, such as condoms and diaphragms, work on the simple physical principle of blocking sperm from entering the cervix. They are the physical walls in our vault analogy. On the other end of the spectrum, surgical methods, like vasectomy in males and tubal ligation in females, offer permanent contraception. These procedures work by physically cutting or blocking the reproductive ducts, ensuring that the sperm and egg can absolutely never meet. While barrier methods use physical blocks, other methods alter the body's internal chemistry. Hormonal methods, like birth control pills, patches, and injections, take a biochemical approach. They introduce synthetic estrogen and progestin into the female body. These hormones trick the hypothalamus and pituitary gland into thinking the body is already pregnant. Because the body believes it is pregnant, it suppresses the release of follicle-stimulating hormone and luteinizing hormone. Without the critical luteinizing hormone surge, ovulation simply does not occur. This means there is no egg available for fertilization, effectively keeping the vault empty. Beyond systemic hormones, there are highly effective localized treatments. Another major category is the intrauterine device, or IUD, which is a small, T-shaped device inserted directly into the uterus. There are 2 main types. Copper versions release copper ions that are highly toxic to sperm, severely impairing their motility and viability so they cannot reach the fallopian tubes. Hormonal versions, on the other hand, release progestin locally. This localized hormone thickens the cervical mucus to create an impenetrable barrier for sperm, and simultaneously thins the uterine lining to prevent a fertilized egg from implanting. To consolidate our understanding of these mechanisms, let us look at this anatomical diagram. Here we see the female reproductive system, including the ovaries, fallopian tubes, uterus, and cervix. Notice the 4 labeled intervention points. At the top left, an arrow points to a cut fallopian tube, representing tubal ligation, a permanent surgical block. On the right, an ovary is marked to show where hormonal methods suppress ovulation. In the center, a T-shaped IUD sits inside the uterus, where it prevents implantation and impairs sperm motility. Finally, at the bottom, a physical barrier is shown blocking the cervix, preventing sperm entry. This visual perfectly summarizes how different methods target specific reproductive stages.
We have now covered the full spectrum of this topic. Let us review the key takeaways. First, contraceptive methods are essential tools that safely prevent unintended pregnancies by intervening at various biological stages, from physical barriers to hormonal suppression and surgical blockage. Second, we learned that the effectiveness of these methods is rigorously quantified using the Pearl Index, which standardizes clinical data into pregnancies per 100 women years. Finally, comparing perfect clinical use to typical real-world use reminds us that human error significantly impacts reliability. Mastering both the biology and the statistics empowers healthcare professionals to guide patients effectively.